again, welcome everyone to today's Gateway to Translation webinar series. Today we're going to have Matt Robinson speaking about microbiome-based therapeutics. My name is Dr. Virginia Pedicord. I am a group leader in the Department of Medicine at the University of Cambridge, and I was told that I should briefly introduce myself as well. So again, I'm a group leader here at the University of Cambridge. And my group specifically works on using functional metagenomics tools to link mouse and human microbiome research. And we hope that by linking these two, we actually have experimental means to get at the cellular and molecular mechanisms by which commensal microbes affect host physiology, specifically immunity. And so at that, it is my absolute pleasure to introduce Matt Robinson today. So Matt Robinson did his PhD at University College London in immunology, and he then went to go work for AstraZeneca for 10 years. And in 2019, he joined Microbiotica here in Cambridge as a vice president of translational research, and then now a senior vice president. And today he's going to talk to us about microbiome-based therapeutics, a clinic-first discovery of IBD, and immune-oncology drugs enabled by precision microbiome profiling. Thank you, Matt.
So today I'm going to give you a brief introduction to the microbiome, particularly how it fits in with human disease. Then I'm going to give you a little overview of the approach that we've been taking here at Microbiotica over the last 2 years. So that general approach, and then how we've used that in two particular programs, one in immuno-oncology and one in ulcerative colitis. So just as a sort of way of an introduction, this slide is really aimed to pick out the complexities here. So the microbiome, so there's about as many bacteria in and on us as we have cells in the human body, give or take. And the difference here is, of course, the cell in the human body has the same genome, but the complexity of the microbiome is considerably bigger. So if you take your gut microbiome, for example, where the vast majority of all those bacteria reside, each person will have 200 odd species of bacteria within their microbiome, and that forms an ecosystem. But those 200 species can form come from one of any 5 to 10,000 different species that exist in the human microbiome in different people. So there's a huge, great variety in between individuals, but even within one's individual microbiome, there's a great complexity as well. And we only function normally with this microbiome because what the microbiome does is it prevents infection, particularly in the GI tract. It helps us break down food, synthesize vitamins, but it also regulates a number of functions in our bodies. So the gut function is very dependent on the microbiome, but also the nervous system, the immune system are very well regulated by that microbiome. So this interaction of these 200 odd species with the human body is really intricate. And what you have to remember here is, so on the right-hand side, there's a phylogenetic tree of species that are in the human microbiome. And what you see here, these are in 4 main phyla. But what we have to remember is when you do a map of the tree of life, these 4 phyla are highly diverse. The differences and the diversity in the microbiome is huge compared to even the eukaryotic species. So they are more different than we are to plants. So this makes understanding that all very difficult, and actually there's been many advances in the field to enable us to really analyze that microbiome with some precision, etc. Sorry, my slides are frozen. There we go. And those associations with the host and the function of the host with the immune system, the nervous system, and the function of the GI tract, that means what we've seen again and again in many indications is actually the human microbiome is linked with disease. And I'm just showing you one example of a paper that came out a few years ago. It was a review looking at this across multiple different diseases. And what it was able to show is the microbiome was more associated with multiple diseases than the actual genome of the host itself. So this is true across a number of GI indications, so ulcerative colitis, Crohn's disease, some cancers like colorectal cancer and prostate cancer, obviously near and proximal to that microbiome, but also diseases such as rheumatoid arthritis, schizophrenia, etc. So the microbiome is associated with a whole huge array of different diseases in the human. And what does that look like? Well, I'm just showing you one example of here what that could look like. And what we mean by this is when you do an analysis of the microbiome of patients, they look different to a healthy population of people. And this is very true of an example. This example I've chosen is NASH, non-alcoholic steatohepatitis. And what you see in this, the images on the left-hand side, what we will do is call up my, is the microbiome of patients is sufficiently different to a healthy normal population that actually in late stage patients with cirrhosis, you are able to just use the microbiome to predict whether that patient has the disease or not. So this means that the microbiome of the patient is very distinctly different to that in normal healthy individuals. But not just that, but these two curves on the right-hand side show that actually within the staging of that disease, the microbiome progresses over time with the disease, and such that the microbiome is more altered the further down the progression of the disease you get. And this is obviously very complicated because it's hard to split out here what is cause and effect, especially in a disease like this, which is so associated with metabolic disorders, that the patient is likely to have an altered diet and a lifestyle. But what you see on the right-hand side here is even when you take lean patients out of this with NASH and NAFLD, what you see is there is a microbiome association that is above and beyond this. So I think this sort of evidence and many other evidence suggests that there is a real link between the microbiome and disease. And what a lot of people have done is gone on to take a diseased microbiome from patients and transferred that to mice, or give mice a disease and transfer their microbiome on. And what you see is that microbiome is partly driving the disease. So although the disease may cause a change in the microbiome, that change in microbiome reinforces the pathology of the disease. The microbiome is also linked with response to drugs, and I've given two examples, one here, one in on the right-hand side, vedolizumab in inflammatory bowel disease. This shows that the microbiome is able to predict whether or not a patient would respond to vedolizumab. And on the left-hand side, a non-GI indication, so in this case, it's ankylosing spondylitis, that the microbiome of the patient also predicts whether or not that patient will respond to anti-TNF therapy. So what this shows us is that the microbiome is linked to GI diseases, but also diseases outside the GI tract and the response to therapies in that. And just for one sort of final completion, how it interacts with disease, etc., is what we know is the microbiome can also metabolize some drugs. So L-DOPA shown on the left-hand side here, and methotrexate, both in Parkinson's and RA respectively. What we see here is the microbiome be able to metabolize the drug, reduces the drug concentration, therefore lowers the patient's response to that therapy. So over the last few years, we've seen an evolving picture where we recognize the microbiome is involved in disease. It's a contributor to that disease, and it's also a contributor to drug response. And on the right-hand side, I've shown a list of diseases. I think this is now out of date, and there's many more diseases, but we're seeing an increasing list of diseases where the microbiome is associated with that disease. And so what we're now also seeing, and to definitely prove that there is some causation here, is we're seeing the emergence of therapeutic interventions. And this has really started, and a process has been around for many, many years, which is fecal microbiotica transplant, which is where you essentially give a patient the stool of a healthy volunteer patient, and that engrafts in them and helps to normalize their microbiome. And therefore, it's being used to treat diseases where the microbiome is really what we call dysbiotic. So a very altered microbiome, and you normalize that microbiome and you help the patient have a better response to the disease. Now, the poster child for this is really C. difficile infection. So on the left-hand side here shows a clinical trial where patients with recurrent C. difficile infection. So C. difficile infection is a GI infection, typically occurs within hospitals, so it's hospital-acquired infection. And you can treat it really well with vancomycin. But what happens when you treat recurrent patients with vancomycin is you see only 30% are cured of the disease. So 70% have a relapse of the disease. However, when you give the patients vancomycin followed by FMT, 80% of those patients are no longer relapsing. So only 20% relapse and 80% are cured. So the reason for this is those functions of the microbiome where it competes out pathogens, so competes out C. difficile, but also the microbiome of a normal person processes our bile acids to get secondary primary bile acids, and these stop the sporulation of C. difficile and prevent its infection. So that's why healthy normal people don't get this, but people in hospital that have a dysbiosis and or are immunocompromised are susceptible to C. difficile infection. And when you give them back a normal microbiome, then they are recovered. The problem with treating them with vancomycin is it further disrupts their microbiome. So when you treat with vancomycin to kill off the C. difficile infection and then give them an FMT, they're able to recover really well. And a company called Seres has now formed a capsule version of this, where they take all the spores within the stool product of a healthy volunteer and give that to patients. And they show this also is hugely beneficial to the patients in preventing reoccurrence of C. difficile in patients that have already recurred. So this is a huge step change in these patients with C. difficile, which is a significant problem in the US and certain other countries. However, there is a challenge with FMT, and that, well, so first I should say, it's not just C. difficile. There's been clinical studies now that shown that it works in inflammatory bowel disease, irritable bowel syndrome. It helps with cancer immunotherapy. It can relieve some of the symptoms in autism, and it can really help prevent toxicities associated with graft versus host disease. And what's clear with most of these conditions where FMT is working really well is they are conditions where the patient's microbiome is really quite dysbiotic. So the microbiome is not normal and you're recreating a normal microbiome. The difference here is probably the cancer immunotherapy, where it's different endotypes of people that respond differently to cancer immunotherapies, and I'll go and describe that a bit later. However, there are challenges with with FMT therapies. One is there's a significant risk of infection, so the donors are highly screened for both lifestyle, and then also the stool product is obviously screened for presence of various infectious agents, but there's always a slight risk that you can transfer an infection. One of the major problems in the field is reproducibility, and that's come out of some science and clinical studies that show there may be a super donor effect, i.e. an FMT from certain donors works better than other donors. And it's been really hard to understand and to pharmaceuticalize that and really, reduce that super donor phenomenon to practice. It's very difficult when you need a certain type of donor. So these conditions that are driven by an endotype rather than a strong dysbiosis. you know, there it can be really difficult to find donors that are able to be the donors. And even in normal situations, we just have healthy volunteers, because the screening is arduous and nobody really wants to play with their poo much. It can be really difficult to get a good supply of donor material. And then finally, what this has sort of led to is quite unregulated use of FMT, to the point now where if you go online, you can order a kit and such that you can do it yourself at home. They do give you silicone gloves if, in case you're, in their words, a bit squeamish. And it is not a nice procedure. They recommend either doing it in the bath or bringing your own large brown towel. So there really is a need to move on from FMT to something that's more pharmaceuticalized. And many companies, our seed included, are trying to do this by taking defined consortia of bacteria or design single strains of bacteria from healthy volunteers and using those to treat disease. And I'm going to show you some data, clinical data from a company called Vedanta and a product called VE303. What they did is they looked at an FMT study in C. difficile infection, and they looked at the bacteria that were engrafted in patients that didn't go on to recur. And they looked at the donors, and they were looking at the bacteria that were engrafting from the donors into the recipients and correlated with a good prognosis in the clinic. They found 8 different species of bacteria, and so they made a consortium of 8 bacteria out of these. They're manufactured and then freeze-dried and put into a capsule, so you just swallow them quite easy, and they get delivered into the gastrointestinal tract. And what you see here, although the effect of the therapy may be a little bit delayed, is really potent at preventing patients relapsing with C. difficile infection, just like the FMT study did in patients. And so we think that the microbiome field is just at the start of a new discovery phase. We think there are many diseases, as I said, that are linked to the microbiome and where therapeutic intervention may be useful. There are now 3 donor-derived FMT-like products that are licensed for recurrent C. difficile infection, 2 in the US and 1 in Australia. I also mentioned earlier, FMT has been shown to be effective in a number of other conditions as well. And I think finally, a number of companies now have got positive clinical data using an orally dosed defined consortium product. Now these are in ulcerative colitis, immuno-oncology, psoriasis, and also the Vedanta data I showed you earlier in C. difficile. So we think the microbiome in the Wild West of the FMT is now moving forward to a point where we can actually start dosing patients, not with random stool from healthy donors, but with the key bacteria that is going to drive clinical benefits in those patients. And so that's where we, as Microbiotica, have been trying to develop therapies in a number of different areas. And many people have tried to define these consortia of bacteria in different ways to identify the right strains to give to people. Typically, most people do it in a phenotypic way. They're trying to screen bacteria for beneficial effects, either in cell assays or in animal models, and then they use those to then develop a treatment. However, in Microbiotica, we think this interaction between the human microbiome and the patient is hugely complex, and each bacteria is part of an ecosystem and changes that ecosystem. The ecosystem as a whole produces many products, bacterial fragments, extracellular vesicles, etc., but also many metabolites that interact with the patient and the human body in multiple different ways. And we believe we're only starting to understand this. So we believe it's a bit like an iceberg, i.e. the mechanisms that we know of, that we clearly understand how the microbiome is affecting the host response, are actually the minority of what's going on. And the vast majority we haven't yet really got a good understanding of. So as a company, we set up this sort of workflow that I'm showing here, where what we did was we've developed what we think is the best-in-class microbiome profiling. So we can profile the microbiome of patients with unrivaled comprehensiveness and precision. And so this precision profiling allows us to identify specific signatures of bacteria within the microbiome of patients. And it's these signatures that we believe hold the secret to how we develop our therapeutics. So what we do is we take, in the right settings, we take the good bacteria out of these signatures and we develop what we call live bacterial therapeutics or live biotherapeutic products. We develop these consortia of bacteria that we are then using to treat patients. The way we do it, we also have the joy of discovering what we think are really exciting biomarkers that might help in clinic to help determine which patients will not respond to therapies. And as we understand in our bacteria, what we're starting to understand is the metabolites and the products produced by those bacteria. And these themselves can also become targets for small molecule drugs for development in the future. However, as you pick any one individual target in these small molecules, what you're obviously not doing is picking up that richness of how the microbiome interacts with the host in multiple different ways, many of which can be beneficial. So the foundations of Microbiotica came from our CSO, Trevor Lawley, who is a PI still at the Wellcome Sanger Institute, and he span out Microbiotica about 7 years ago. Trevor has been, at the forefront of microbiome science for many, many years now. And one of the things that Trevor is brilliant at, he's a fantastic microbiologist. And so what Trevor's been able to do is culture out many of the bacteria that were previously thought to be unculturable. And so what we've done is we took this into the company and we industrialized it. We formed a process called personalized bacterial banks, where we take the stool of a patient, the microbiome of a patient, and we culture out just about every single bacteria and isolate every single bacteria within somebody's microbiome. And we've done this across many, many different individuals, some healthy volunteers, some diseased, some with various diseases. We've done it across 44 different countries now. And so what this gives us is a culture collection, a phenomenally huge culture collection of human bacteria, commensal bacteria. So these are a library of potential therapeutics, but they're also a source of genomes within the microbiome. So what we do is we pull these forward and we isolate and sequence the genomes of many of these bacteria. And when we understand the genomes of these bacteria, it enables us to do a process called shotgun metagenomic sequencing. And we use these genomes as a reference database when we do that shotgun genome sequencing to map which bacteria are present and absent within a microbiome. And the bigger and the more comprehensive your database, the smaller the dark matter or the unrecognized bacteria in there. And on top of this, what we have is what we think is a leading bioinformatics pipeline tool and machine learning tools to really understand the bacteria in a complete sense, in a microbiome, in an ecological sense, and how that differs between patients. So what we believe, we have a unique asset that really gives us a global microbiome blueprint. And what does this mean? Well, this gives some data where we tried to benchmark how we profile a stool sample. On the left-hand side here, this shows so in a shotgun metagenomic sequence, how many bacteria we can identify what they are. And what you see when we compared us, we can identify over 95% of the bacteria that are there. And when we compare that to a commercial provider that was using the publicly available database, they can only identify about 50% of the bacteria that are present there. Now, this is a little bit old, so they're probably a bit better, but it's probably still up to 70%. So Still, there's a large proportion of the microbiome that are not understood by many people. But with our reference database, we can do that. And not only that, but when you look at these reads and how they classify against the genomes, when we look at our reads, they are well over 95% similar to the genome that we identify them as the species are, where if you deal with that with the public databases or on commercial providers, you drop below this 95% threshold. This 95% threshold is really important because when you look at genomes of different bacteria, bacteria of the same species, you say, have a 95% identity in their genomes. So what we're showing here is not only do we identify more bacteria, but we are doing it with it at the species level and below the species level, where many others struggle to get it below the level of the genus. So that describes to you how we built a platform to enable us to analyze the microbiome of patients better than anybody else and have a culture collection of bacterial strains that we could use as therapeutics. Now I'll show you two programs where we've put that into practice, and we're about to go into the clinic in both programs to treat patients. So the first program is Immuno-oncology. I just want to introduce checkpoint inhibitors and how they changed melanoma treatment over the last 10 years. So about 10 or so years ago, most patients with melanoma would be treated with surgery. This is successful in most patients. However, patients where it had advanced or was malignant had a very poor prognosis. These patients would have it surgically removed, but then go on to chemotherapy. And what you see from this Kaplan-Meier plot is actually the disease would progress in the patients very rapidly after chemotherapy, such that within 3 months, 2/3 of the patients, their tumor was back growing. Within a year, 95% of the patients, their tumor would be back growing, and the prognosis for 2-year survival was about 5%. Immune checkpoint inhibitors came along. So tumors express checkpoint inhibitors on the cell surface, and this interacts with receptors on T cells, cytotoxic lymphocytes in the human immune system, and it turns them off so they can't kill the cancer. However, if you block these receptors on the T cell with an antibody called a therapeutic antibody against these PD-1 receptors, it is like the T cell no longer sees this cloak of the ligand that the tumor expresses and is able to kill that tumor cell. And this has revolutionized treatment of melanoma. Now, when a patient comes in, when they're treated with pembrolizumab, 40% of those patients will not have progressed in the first year. And we now have the data that in 5 years' time, this 40% patients will still be alive. And this is truly long-term survival. These patients are surviving, and it essentially looks like a cure. So this is very exciting for melanoma, and it's transformed a disease that was a death sentence. But the hunt in the field now is for an ability to change these patients that are not responding to the therapy to patients that do respond to the therapy, because the non-respondent patients still have this very poor prognosis where they next take chemotherapy as they move forward. Now, what was amazing to me as a non-microbiome scientist at the time, when I worked in the immuno-oncology space, is 4 publications that came out more or less at the same time that showed the microbiome of patients before they took this checkpoint inhibitor really was associated very tightly with whether or not they responded. To the microbiome field, this was not surprising because we know that microbiome is very linked to how the activity of the immune system. So the microbiome of patients changes the rheostat of the immune system, and then this anti-PD-1 therapy takes off that specific inhibitor of the immune system at the tumor. But this wasn't just an association, this was causation. So when you transferred the microbiome to mice, you transferred their responsiveness to anti-PD-1, but also some later studies that came out after we started this, when they took the microbiome of patients that had had a complete response to anti-PD-1 and transferred that to patients that had failed anti-PD-1 therapy. Some of those patients, when they took anti-PD-1 again, then became responsive. And I'll show you scans from a patient here that had a melanoma metastasis in the leg. And as they were treated with anti-PD-1, what you see actually in immunotherapy is it lights up a brick lighter. And this is the immune response fighting the tumors. And eventually these tumors are completely gone from the patient. So by changing the microbiome of the patient, you can completely change their response to immune checkpoint inhibitors. The challenge here was all these studies that I showed you all found a different series of bacteria that were associated with this response to immune checkpoint inhibitor. So this created a lot of doubt in the field, but also when you're looking at a clinic first approach, it made it very difficult to decide how we could move forward and which bacteria were the right bacteria to treat patients. So we brought to this our own study that we did in collaboration with Pedicord and a team at Cambridge University hospitals, where we looked at melanoma patients taking immune checkpoint inhibitors and followed them over time. And the other thing we brought is, of course, our platform that gave this high resolution of the microbiome of patients. And because our platform can be used on any shotgun metagenomic sequencing, what we did is, because we wanted consistency across multiple data sets, we took those 3 original publications that were in melanoma and combined those with our own study to find a signature that was associated with response to checkpoint inhibitors across multiple cohorts. And that's exactly what we did here. So this graph on the left-hand side, each dot is a patient, and where they are on this y-axis is how well they or how poorly they will respond to immune checkpoint inhibitor. So the higher the dot, the less likely a patient is to respond to therapy, and the lower the dot, the more likely they are to respond to therapy. But just based on a prediction for the microbiome, based on a signature of bacteria. And what we've done here is split the patients into non-patients that then went on to not respond and patients that respond. What you see here, we have this association of the microbiome response to checkpoint inhibitor in melanoma. And as I said, this is across multiple cohorts. And the key for us was to find a signature that works across multiple cohorts. And you can see it works very well across the combined data set, but across also each of the individual data sets individually. So we've got cross-cohort comparison that worked, that we validated both in melanoma cohorts, but also in other indications like non-small cell lung cancer and renal cell carcinoma. So we've got this signature we think is very robust, works across multiple indications. But here we show a volcano plot showing the different species in that. And what you see here is the further you are to the right-hand side, the better the bacteria. So the more you are associated with a positive response. What you see is the bacteria in the signature dominated with bacteria that are beneficial for response to immune checkpoint inhibitors, and i.e. raised in patients that respond. We focused on these 9 bacteria because they alone were productive and predictive of response. But also, it's worth noting that in these 9 species, there were 4 novel species. So these were species that have not been identified by anybody else outside our company. And we now pulled these together, make a consortium of these 9 bacteria that we are now developing as a therapeutic. It works in mouse models. So this is a tumor growing on a mouse. When we treat it with our therapy, the tumors don't really grow any slower. We see this marginal decrease in tumor growth, but nothing to the effect that you see with an anti-PD-1 therapy like we treat with patients. However, we see synergy when we put the 2 together and we see them together. So we're also doing lots of studies to understand how these bacteria be having this effect. What we know is to drive a tumor cell killing by cytotoxic T lymphocytes, they first need to be activated and primed by dendritic cells. And dendritic cells are one of those cells in the immune system that interacts directly with bacteria and drives that T cell response. So when we looked at all our bacteria individually and as a consortium and compared to them 3 of the most potent immune stimuli that we know, our bacteria drove a really strong dendritic cell activation, producing lots of a cytokine called IL-12 and less of a cytokine IL-10. IL-12 drives cytotoxic T lymphocytes. IL-10 inhibits them. So what we are driving is a dendritic cell that's likely to activate cytotoxic T lymphocytes, these key immune cells. When we combine these dendritic cells with these T cells, we do indeed drive a strong activation of the T cells. And then when we take them and add them to tumor cells, they are very potent killers of these tumor cells. So we think this is a key mechanism of how our bacteria are driving this response that we see in mouse, we see in association in patients, but they also drive strong activation of NK cells, another cell type that is able to go on and then kill tumor cells. But we also started to discover new mechanisms. Some of our bacteria interact with macrophages in the tumor microenvironment that inhibit the T cells in the tumor microenvironment, and we show that metabolites from our bacteria are able to reverse this inhibition. So we're really excited by this. We're starting to understand new mechanisms of how the microbiome interacts with patients to drive this CTL response and this anti-tumor response. Our bacteria all look perfectly safe with a highly screened, a highly profiled. They have the profile that's very consistent with normal, prevalent human commensal bacteria, which is they are. They are screened for developability. We have manufactured each of these at scale, and we now have GMP product of all of these bacteria. And we're about to go into clinical trials over this summer, where we will be taking melanoma patients that have failed to respond to anti-PD-1 therapy. And then we will be treating these patients with PD-1, as they did in the FMT study, plus our therapy to see if we can convert patients that do not respond into patients that do respond. This study has two arms. One arm, we're going to give the patients vancomycin. This is just to create a niche within the microbiome of the patients to allow our bacteria to engraft a bit more. What I'm really excited about, what's really nice here, when you look over the whole study, we discovered this bacterial signature in multiple trials in melanoma, taking the therapy. We identify them, we test them, we manufacture them, and we're about to go back into exactly that same population of patients that have a negative microbiome that is preventing them to respond to checkpoint inhibitor therapy. And just one other thing, as a company, we're not stopping there. We've extended our collaboration with Cambridge University Hospitals to one with Cancer Research UK as well. And we're now doing a much bigger study across multiple different tumors, across multiple different treatment effects, just to really understand, is that microbiome similar between the different indications and the different combination therapies that checkpoint inhibitors are used in. So we really can really understand at a deep scientific level how different microbiomes interact with different patients. I will hurry up because we're running out of time, just to take you through our UC program. This is an entirely independent study. What's key for us is that really good quality clinical data to start our analysis with. And the rationale for why the microbiome affects inflammatory bowel disease, and particularly UC, has been developed again over many years, where they show patients with UC and different forms of inflammatory bowel disease really have an altered microbiome from normal patients. When you transfer this microbiome into mice, what you see is those mice get a colitis type pathology. And also in the clinic, what we knew when we started this program is multiple people have shown when you take mild to moderate patients with UC and you give them FMT, a good proportion of those patients go into complete remission. So on average, about 30% across these different studies. And as I said, what's really important to us is really good quality clinical data. So one of those studies was run by Sam Costello down in Adelaide. Sam is a brilliant GI doctor, but he also really understands the microbiome. So he prepared his FMT anaerobically and kept and preserved those bacteria. This gave us the best chance to do high quality analysis, but also meant that he got really great response rates in his patients, with the response rates in his patients similar to that we see on many therapies like anti-TNFs. So what we did here is we took the healthy donors and the patients before they received the FMT and after they have responded or not. And what we did is we did deep microbiome sequencing of the healthy donors and the patients pre and post FMT. And what we did, we were able to analyze these patients and look at the bacteria that had engrafted from the donor into recipients and how this correlated with response. So we designed a really cool bioinformatic tool, and this shows the abundance of a species of bacteria in patients, and this is the healthy donor. This is the abundance of the species in patients before the FMT, and then this is the abundance in the donors after they take FMT. If the lines are red, it means that there's a worsening of the condition. So obviously, as you go from donor to patient, there's a worsening condition. And what you see is where this bacteria is engrafted, many of these patients have had a beneficial effect, i.e. the lines go green. So what we did is we built a bespoke tool where we did the analysis and we looked at what happens when you take one of these species, how predictive is it, how associated with a good response. And then if we mix two of these good bacteria, are they even more associated with a good response than you would expect? And what we did is we did this multiple times and we came up with a consortia, 8 different bacteria that represented species that were consistently associated with a beneficial response when they engrafted into those UC patients. So again, we wanted to understand how these bacteria work. Can't just take them in the clinic. We need to understand the bacteria work. And when we think of pathology of UC compared to healthy individuals, I think in terms of host biology, that the intestinal, the epithelial barrier is damaged, which causes leakiness of the bacteria within the submucosa. What you see is an aberrant response of the innate immune system, so the macrophages, and again, the dendritic cells within patients. This abnormal response to dendritic cells then drives, again, an exaggerated T cell response, all of which gives a pro-inflammatory cytokine milieu, driving inflammation in the patients. And when virtually all the therapies that we know and use in IBD are systemic therapies that just block part of this inflammatory cascade at some point in the process. Whereas we wanted to look at how our bacteria affect all parts of this disease pathology cascade. So I'm just showing you highlights of some bacteria in some assays because it gets very complicated. We tested all of the 8 strains of bacteria across multiple assays. None did anything bad. But what I'm showing here is one of our strains was particularly good at improving the barrier integrity of a gut epithelial bilayer. But 3 of the strains were really, really good, not just at doing that, but what they did is if you damage that epithelial barrier with LPS, like an inflammatory insult, it helped repair that barrier. And this is where we've really been able to make great progress understanding what the mechanisms of these bacteria are. So we know the metabolites that 2 of these bacteria release that drive these beneficial effects on the epithelium. And we've also identified a completely new novel protein expressed by one of these bacteria that drives this beneficial effect. Our bacteria interact with these innate immune cells, the macrophages and the dendritic cells, and they skew the immune response. Again, looking at cytokines, here they induce lots of IL-10, this immune inhibitory cytokine, but they don't affect the TNF production from these cells. So TNF is a pro-inflammatory cytokine. So this is driving a more anti-inflammatory profile of that innate immune system that is the part of the immune system that directly responds to the bacteria. And then what I told you is dendritic cells typically drive an immune response, a T cell response. What we show is when we take these dendritic cells treated with these bacteria for 310, instead of driving an inflammatory immune response, what these do is they actually make the other arm of the T cell response, the CD4 T helper cells, instead of making them pro-inflammatory like we saw with the other bacteria, These are anti-inflammatory because they are producing IL-10, which is typical of a regulatory T cell as instead of an inflammatory effector T cell. So what we really think we're doing here is this is a different type of therapy that we're bringing to this patient population, not one that you give systemically and blocks the immune response in patients. What we're doing is we're bringing a therapy to these patients that interacts with the host in multiple different ways through multiple different mechanisms to intervene at many important nodes within the inflammatory pathology of this disease. And then this is my final sort of information slide. So what we see here is we're actually taking this then into patients, and this is a randomized controlled trial where we are comparing patients that taking their standard of care, mild to moderate UC patients, so active disease that's inhibiting their everyday life. These patients with this active inflammatory disease, we're going to treat them either with placebo on top of their standard of care that they're already receiving, or MB310 on top of their standard of care, and see if we can drive some of these patients into remission, as we saw in the FMT studies. Bearing in mind, instead of just giving them a random slew of bacteria from patients, what we are now doing is concentrating on the active parts of that FMT, and we are able to dose this in patients every day to keep that dosing up for 12 weeks. So we're really optimistic that we should drive a really beneficial effect out of these patients with this therapy. So what I want to show here is I think microbiome is a really exciting area in therapeutic research. There's a lot of activity on the area, people developing different consortia of bacteria, different products in different ways. We really believe in this clinic first approach because we think it captures a lot of the biology there that we don't fully understand. We believe from this approach, we've made two really exciting therapies that are helping us, that we think will be beneficial to patients, but are really also just helping us understand some new biology of how the microbiome interacts with patients. And going forward, we're hoping to take both of these into clinical trials later this year. And we believe this is really an exciting area, and we're hoping for many beneficial results in the near future. So finally, my thank you slide. I'm really running over, so apologies. Many great collaborations with Pippa and her team, and Dave Adams and Sam Costello at various institutes. Trevor, our CSO, set us up. Tim and Mike were our CEOs. But really, everybody at Microbiotica had to be involved in helping to deliver these therapies to patients. It's an extremely complicated area of biology, from our microbiologists, our bioinformaticians, our translational biologists, our preclinical development people that are able to manufacture these really hard to grow anaerobic bacteria, and our clinical staff here as well. So there's a big thank you to those, and thank you to all the patients that were involved in the studies that allowed us to develop our products, and obviously their families that are in a vulnerable situation.